Greetings. Myself, N. Usha, Department of Microbiology, Sacred Heart College, Autonomous, Tirupattur. In this video, we have to see about the ultrastructure of bacteria. First, we have to see about the general characteristics of the bacteria. So, bacteria are the unicellular organisms. Bacteria are the microscopic single-celled organisms that will be measured in micrometer. They lack the cell organelles such as the chloroplast and mitochondria, and they do not have the true nucleus. The cell membrane and the cell wall are referred to as the cell envelope. Asexual reproduction by binary fission. However, some bacteria can also exchange the genetic material among one another in a process known as a horizontal gene transfer. This is the ultrastructure of the bacteria. The outer layer is a capsule. The center layer is cell wall that is present between the capsule and plasma membrane. The inner layer is a plasma membrane. The folded structure is called as mesosomes. The innermost layer is a cytoplasm and that will be contain nuclei, DNA, flagellum, plasmids, inclusion bodies, polyribosomes and phylers like that. So in this we have to see about the bacterial cell structure. Cell envelope that consists of cell wall, plasma membrane and capsules. The cell appendages that will contain flagella, pili and fimbriae. The cell organelles that will contain nuclei, ribosomes, mesosomes, cytoplasms, inclusion bodies and plasmids. First, the cell envelope that contains the cell wall. The cell envelope is composed of the cell membrane and the cell wall. The bacterial cell wall is provide the structural integrity to the cell. The peptidoglycan molecule found only in the bacterial cell wall. The peptidoglycan is made up of the polysaccharide backbone consisting of alternating N-acetyl muramic acid that is NAM and N-acetyl glucosamine that is NAG residue in equal amount. The bacterial world divides into two major classes. First one is gram positive and another one is gram negative. The amount and location of peptidoglycan in the cell wall that determines whether a bacterium is gram positive or gram negative. So gram positive cell wall are the thick and the peptidoglycan layer is constitute almost 95% of the cell wall. It takes up the crystal violet dye and are stained in purple color. The gram negative cell wall are much thinner than gram positive cell wall and they contain a second plasma membrane and they are stained referred as a pink color. Next one is plasma membrane. The plasma membrane also called as a cell membrane or cytoplasmic membrane because the plasma is another term for the cytoplasm. The membrane found in all these cells that separate the interior of the cell from the outside environment. The plasma membrane functions provide the protection for the cell. It also provides a fixed environment inside the cell. Next one is capsule. The capsule is a layer of slime that lies the outside the bacterial cell wall. It's secreted by bacteria and diffused into the surrounding the medium. That functions. Capsules have this significant role in determining access of certain molecules to the cell membrane, mediating adherence to the surfaces and increasing the tolerance of the desiccation. Next one is cell appendages, that is flagella, that contain flagella, pili and fimbriae. First one is flagella, the bacterial species that differs in the number and arrangement of the flagella on their surfaces. The flagella beat in a propeller-like motions to help the bacterium to move towards the nutrients, away from the toxic chemicals towards the light, that is photosynthetic cyanobacteria. So bacteria may have one, a few or many flagella in different positions on their cell wall. First one is monotrichous, the single flagellum, amphitrichous, a flagellum at each end, lophotrichous, clusters of flagella at the folds of the cell, peritrichous, flagella distributed over the entire surface of the cell. Next one is pili. The pili are made up of the protein subunit is called as a pilin. The range in length from a few fractions of a micrometer to over 20 micrometer. Pili are hair-like structures on the surface of bacteria. Functions. Pili help them to attach the surface of bacteria, mediate the cell-to-cell interactions and more. Next one is fimbriae. So fimbriae are made up of protein subunit is called as a fibrillin, range in length from 4 to 35 nanometer. Fimbriae are finger-like projections. Functions. The fimbriae help them to attach the surface of bacteria, mediate the cell-to-cell interactions and then more. Next one is cell organelles that contains nuclei, ribosomes, mesosomes, cytoplasms, inclusion bodies and plasmids. First one is nucleide. Unlike the eukaryotic, that is true cells, bacteria do not have a membrane enclosed nucleus. The nuclei is a region of cytoplasms where the chromosomal DNA is located. It is not a membrane bound nucleus but simply an area of the cytoplasms where the strands of DNA are found. Next one is ribosomes. It involves in synthesis of protein. Number varies with rate of protein synthesis abundant in cytoplasms, often grouped in long chains called polyribosomes. Bacterial ribosomes categorized into two subunits. Bacterial ribosomes have sedimentation rate of 70s. Their subunits have rates of 30s and 50s types. Next one is mesosomes. In bacteria, depend on growth conditions, mesosomes appears to be enfolded at more than one point, such as inholdings called as mesosomes. Mesosomes are two types: central mesosomes, peripheral mesosomes. Central mesosomes located near to middle of the cell. Peripheral mesosomes are not restricted to middle of the cell. It forms cross wall during the cell division, cause the cell division of cell. Next one is cytoplasm. Cytoplasm of the bacterial cell is a various watery solution called cytosol that containing a variety of organic and inorganic solutes and ribosomes, containing plasmids in some species of bacteria, not containing endoplasmic reticulum, membrane bearing microsomes and mitochondria. Next one is inclusion bodies. So inclusion bodies and the cell inclusions are both abnormal structures that can be found in these cells, but they differ in their composition and function. It helps to store the energy, carbon and other substances. The final one is plasmid. The plasmid is small circular double-stranded DNA molecule that is separated from a cell's chromosomal DNA, and it is extra chromosomal DNA. So plasmids are found in the bacteria and some other microscopic organisms. They can be passed from one cell to another and replicate independent of the bacterial chromosomes. Thank you.